for advancing sciences. And so using the ever-increasing power of computers, we can model mechanisms, we can analyze and predict potential outcomes, and analyze immense data sets. So in our presentation today, we'll be focusing primarily on the idea of analyzing and manipulating large data sets obtained from whole exome and whole genome sequencing runs to identify mutations and variations from the norm. Because of both of our backgrounds in cancer biology, there is going to be kind of a cancer theme to it for the presentation, but the idea is nonetheless the same for any WES or WGS data sets when you're looking for variations from the norm. So those who are interested in cancer, you can literally go home and start looking for your own mutations and make findings right after this workshop with a bit of know-how. So this tutorial focuses mostly on using Unix, so Mac OS or Linux command lines. If you're using a Windows machine, the commands will be a little different, but the theory remains the same. You can easily find like the equivalent commands to those found on for Unix and Linux. On Windows, you can find them online, or you can install a Linux environment on your Windows machine with some know-how. This could be a little complicated, but it can be done. So I'm sure you guys have seen people on this cool-looking black display frantically typing and scrolling. This might look like very advanced computing techniques, but in reality, it's actually very, very simple. So what is the terminal and the command, or the command line, in other words? The command line or terminal is a text-based access to your operating system. So here on the screen, I have two images showing the exact same thing, but one is using the graphical interface we all know and love, and the other is using the terminal. We can see here that we're in a folder called articles. A folder called articles, also known as a directory. Folders can also be called directories. And within this directories, we have all my saved files. For example, we have characterization of gastric adenocarcinoma. So as you can see, it's the exact same thing on both screens. One is graphical, one is command line. So how do you navigate using command line? So here are some basic commands that you can actually use on your command line. First, we have the command cd and cd ... which stands for change directories. So this allows you to move in and out of directories or folders. So to enter a directory, you would type cd followed by the name of the directory, and you'll press enter, and this is going to allow you to go into that folder via command line. To back out of a directory, you would just type cd space ... press enter, and this is going to bring you back out to where you were previously. Next, we have the ls command. This stands for list. Typing ls and pressing enter will list all the files present in your directory you're currently in. I forgot to mention, if you guys have your command line open, you can try this out as we do it, and I will be doing live examples as well as we move forward with the variant calling stuff. If you do encounter errors or it doesn't work, feel free to email me later. I already sent my email address. So next we have the pwd command, which stands for present working directory. So this command will show you the path to the directory you're currently in. So how did you get to where you are right now? What folders did you go through to get to the specific folder you're in? And finally, we have the vim command. So this is a text editor command. There are other text editor commands you can use, such as nano or emacs. I just prefer using vim because that's the one that I was taught, but they all generally do the same. By typing in vim followed by the name of the file you want to read, you will see what's written in the file. Of course, you can't vim a folder because folders are not files. They contain other files. But if it's a file like a Word document or a note, you can vim it and you can read everything that's inside. So if both the graphical interface and the command line are the same thing, why would you use one over the other? So using the command line is less demanding on your computer because it doesn't have to generate the graphical images that you do see. Also, using command line allows us to navigate and manipulate directories that are not available via the graphical interface. Finally, many tools used in bioinformatics don't have graphical interfaces or GUIs and can only run on command line. And this is for the majority of bioinformatic tools have to be run on command line. Most importantly, though, command line looks really cool and you can tell your friends you're a hacker. So now that we have the basics of using command line, we can go into the more important stuff. So that's somatic variant calling. Again, so this is only one branch of bioinformatics looking at mutational data. There's plenty of stuff you can investigate, especially once you get the hang of the basic commands. So in other words, the mutations present in samples, so somatic variant calling is looking for the mutations that are present in the samples that we have sequenced through next generation sequencing. Because of time constraints, I will only go through the first two major steps of the pipeline. And when I say pipeline, that's your protocol for the bioinformatics. And doing these first two steps will already allow you to visualize some of the mutations that you might have in a data set, so in a sample. So here are the required programs for the full protocol. That is all the steps. But for our purposes, we will only need the BWA tool, Samtools, IGV, a reference genome, and NGS data. For those who didn't see the document, I uploaded on the Slack with links to download all of these. The reference genome and the NGS data I've also put on the Google Drive, and the link is on the Slack. So you guys can do that on your own time or do it right now if you want. So to understand how these programs work to get the mutations, we need to first understand how next generation sequencing really works. And so sample DNA is first fragmented, and the fragments are then cloned and amplified to generate many copies. Each fragment is known as an insert. These inserts have adapters and barcodes ligated to their ends for downstream processing. The collection of inserts are known as, from a single sample, will be known as a library. And so these libraries are then loaded onto lanes of a sequencer, and then the sequencer will read the genetic code of the DNA. So once the sequencer reads the genetic code, it will output what's known as a FASTQ file, which will contain all the reads as well as their quality score. So how well did the sequencer actually read that base? I'm going to get into this a little deeper as we move along. These are files, these FASTQ files, we will process through our variant calling pipeline to see what the mutations are, which mutations are present. So this is where our work begins as bioinformaticians and computational biologists. So the FASTQs will be aligned to a reference genome. Using BWA. This will generate a new file called a SAM file, which we will then sort and convert to something called a BAM file. And again, I'm going to get into this. If this sounds crazy, don't worry about it. I will get into it. The BAM files are then filtered, recalibrated, annotated through the whole variant calling pipeline, such as GATK, but there are other variant calling pipelines that you can look at, or protocols that you can look at. And we end up with what's known as a VCF file, which will show you what mutations are confidently present, and it will tell you whether or not they actually have an effect in the cancers that you're looking at. This might seem very straightforward, but if you just look at this like little diagram that I did, it seems like it's a very short kind of procedure. But like any experiment, most of the times we end up looking like this, and that's because you're going to encounter errors, and the errors are going to keep coming, and you're not going to know why the error is there, and you're going to have to do research and read literature to figure out why the program is not moving forward. So this requires a lot of troubleshooting, but eventually you set up your pipeline and you're good, and you can script it and it will run on its own, clear of errors. So Firstly, the FASTQ files contain the sequences read by the sequencer, as I mentioned earlier. So there are 4 lines in a FASTQ file per read. So the first line is the sequence identifier, which will usually list the machine used, the run number, the flow cell ID, everything that has to do with the sequencer will be listed in this first line of the FASTQ file. The second line, um, the second line of the FASTQ file is the actual read that the sequencer read. So those are the nucleotide, the string of nucleotides that the sequencer read. The third line is just a spacer, and the fourth line is your base quality calls, or the quality of each base called by the sequencer. And so each these are usually encoded by the sequencer, which is why it looks like gibberish. Yeah, to make it, and encoding it makes it easier on the computer to read and requires less memory, which is why they encode it. But there are lists to convert this that can be found online. These quality scores can also be converted into what's known as Fred scores, and which is an integer representation of the probability that a base call was an error. So let's do a little example here. If a certain base has a Fred score of, let's say, 10, that means there's a 1 in 10 chance that that call by the sequencer was incorrect. Another way of interpreting this would be to say that there is a 90% chance that that base call was accurate. If the Fred score was 20, the probability of an error would be 1 in 100, and so on and so forth. So what can we do with these FASTQ files? Well, the FASTQ files have all the sequences that were read, as I mentioned earlier, but these need to be aligned to the reference genome so that we can figure out what the differences are between the two. And once again, BWA is the tool that's going to do this and align it to the reference genome. So there are 3 main algorithms in the current version of BWA, which are BWA SW, BWA backtrack, which is composed of BWA ALN, followed by BWA SAMP and SAMC, depending on if it's single end or pair end, and finally BWA MEM. Each of these algorithms have its own use. For example, BWA backtrack is generally considered better if the reads you get from the sequencer are smaller than 70 base pairs. BWA SW and BWA MEM are very similar and are used if aligning reads that are over 70 base pairs to 1 MB. So BWA MEM is the current standard for these types of reads. So the algorithm basically uses what's known as an index reference genome, which I'm going to explain later, to identify all the regions within the reference where the read can align to. So now we'll take a bit of time and we'll do the live example for those who are following. We're going to run the program right now. So I'm going to open my terminal here. So if I do ls, you can see I have I have my FASTQ file there. So what I'm going to do is we're going to run BWA. So normally, if you type BWA and you press enter, it's supposed to activate the program. However, you can see it here, it says command not found, and that's because the command is not in your path. So the computer doesn't know where to go to find the program. And so we need to add the program to our path. And so to do this, you would type export. And again, this might sound crazy or look crazy, but eventually you just remember these on your own and you won't have to look it up. But so you would type export path, and capitalization does matter on the command line. So it has to be typed exactly like this, equals $ path colon, and you need to put the path to where the BWA program is, so where you saved your BWA program. So for me, I have this other tab open. I'm on my desktop right now, or I'm one level before my desktop, and I've saved it in this file called software. And so to go in, I'm going to do cd space software, enter. ls. Now I'm inside my softwares. As you can see, I have multiple softwares. Again, cd bwa. Whoops. bw, enter, ls. So now I'm in my bwa folder. So I need to know, how did I get to this file so that I can tell the computer how to how to access the program. And so to do that, we have the pwd command, as I mentioned earlier. And so this right here is the path to my program. So I'm going to copy that, switch back to my other tab, paste it. And so now I have added the path to BWA. I'm going to press enter. Now, if I type in BWA, enter, you can see the program was called. So now the program is active, and you have all the different options that you can use. So for our case, we're going to use the BWA mem. So I'm going to type in BWA space mem, enter, gives you all the options that you can run for BWA. For our case, so we have all our options here. You can see it even tells you how to use it. So BWA mem, whatever options you want, your index, your FASTQ, and then we're gonna write that to a file. And so let's do that right now. We're gonna do BWA space mem. We're going to give me a second. Yeah. Okay. So we're going to do BWA mem. We're going to put our indexed reference genome, which I've sent to everyone. And again, we need to put the path to that file. So I'm going to go back. I'm going to find where I saved my So I'm going to go to where I saved my reference genome. For me, it's saved in this folder called genome, right there. So I'm going to pwd. I'm going to copy that. You'll see. I'm going to come back onto here. /hg19, that's the name of the file. Space, now I need to put the FASTQ. So for me, the FASTQ is called, well, for everyone, it should be called like this. We're gonna put this little greater than sign, and then we're gonna call the next file with the same name .sam, and we're gonna press enter. And so this has now activated the program. You can see it's starting to align the reads. This generally, depending on the size of your data set, can take a very long time. I specifically chose a very small data set so that it takes a short amount of time. So while this runs, we can get back to the presentation. Yeah, so the human genome, as you know, is very large and has many repetitive sequences. A single read could potentially map to multiple areas of the genome. So how does BWA algorithm know where to map such repetitive reads? So when the algorithm encounters multiple mapping regions for a read, it will take into account the number of mismatches and the base quality scores that were initially given by the sequencer. Only looking at the number of mismatches isn't enough to choose the right mapping region. You can imagine a scenario where the nucleotide pair so you can imagine a scenario where, although there are two mismatches in the second read over here, the read quality of this TC is actually extremely low. That is, it has a high probability of being a sequencing error, but the base quality score of this A over here can be very high, suggesting that it was properly aligned. So if only looking at mismatches, the algorithm would have mapped the read to chromosome 10 since it only has one mismatch. However, if you take into account base qualities, this TC over here can just be a mutation that was present in our samples. And therefore, the program has to take in both mismatches, and base quality scores for a read, and it will assign what's known as a mapping quality, or it will assign a mapping quality. The higher the mapping quality, the higher the probability that the read maps to that area. So in this case, the algorithm would have mapped, would have taken the base quality scores and the number of mismatches and has assigned a mapping quality of 10 compared to 1, suggesting that the read does actually align to chromosome 10. If there are two regions to which a read can map and they both have the same mapping quality, the aligner will by default map these ambiguous reads randomly to a location and assign a mapping quality of 0. So this can actually be modified to map to multiple locations, but nonetheless, we don't want mapping qualities of 0 because these are ambiguous and we don't know if they actually go there. So there are ways to reduce the number of ambiguous reads by choosing the method of sequencing that's done in the first place. So sequencing can be done in two ways. It can be single-ended or pair-ended. So in a single-ended approach, the DNA library is read from 5' to 3' in one direction only. And this approach is usually cheaper, but if there is the presence of repeat sequences, there is a possibility of getting these ambiguous reads. So to avoid ambiguous reads, we have paired end sequence. We paired end sequence the library. So in paired end sequencing, the DNA is sequenced from one end to a specified length and then resequenced in the opposite direction. The reads from the sequencer don't span the entire length of the insert fragment that were made during the library prep. So if a read falls within the confines of a repetitive sequence, like you see in these red bars, we have ambiguous alignments. So BWA will not know which red bar is the correct one to align the red to. However, if we do paired end sequencing of the same insert from both sides, we can see that the opposite side of the read aligns to the green area, which allows us to map the read to the proper area on the reference genome. So by doing this, we remove ambiguous reads and identify inversions and confirm deletions, et cetera, et cetera. So for a real life example, we can see here that we have reads that align both to chromosome 16 and to chromosome 3. If we only did single end sequencing, if we only did single end sequencing, we would not know which one of these are correct. If we did paired end sequencing, however, we can now see that the insert aligns much more closely to chromosome 16 as opposed to chromosome 3. So that removes the ambiguousness of the alignment. So getting back to the pipeline, the output from BWA is what's known as a SAM format file, which stands for Sequence Alignment Map Format. This file is human readable in that if you were to vim the file or use a text editor, you would get the data which you can interpret yourself. However, attempting to read a whole SAM file is very demanding on your computer, so it's better to just see portions of the file, which can be done with another command called the head command, which you can look up later if you'd like. So if you did, however, vim your SAM file or read portions of your SAM file, what you will see is a list of characters like this, which at first might look nonsensical, but actually makes a lot of sense. Each one of these boxes delineates a read and its mapping to the reference genome. So here's how you interpret the data from a SAM file. First, we have the read ID, which is straight from the FASTQ, the same stuff that was there in the FASTQ that I mentioned earlier. Then we have what's known as the SAM flag. So this number indicates different attributes of the read. There are tables available online that explain to you what each number means. In our case, we have the number 0, which means that the read was mapped to the forward strand of the reference genome. Then we have the chromosome and the chromosomal position to which the read was aligned to. Next, we have the mapping quality of the read, followed by what's known as the cigar string. So the cigar string tells you how your sequence aligned to the reference. So let's take this example on the right. We have 2S, 4M, 2D, 6M, 3S. So 2S means that the two first nucleotides have been what's known as soft clipped. This indicates that these nucleotides did not align to the reference, but the program has removed or clipped these nucleotides because the alignment for the rest of the sequence has high confidence. And so this is only done at the start and at the end of sequencing, where the error rates are much higher than randomly in the middle of a sequence. Then we have 4M, which means that the next four nucleotides have matched the reference genome, followed by 2D, which means there is a 2 nucleotide deletion at those positions. Then 6 more matches and 3 more soft clipped nucleotides. So the cigar string is really just a quick way to know how the alignment looks for that specific read. And you can scroll through all your reads and check all your cigar strings to see if there are any mutations. However, with millions and millions of reads, this will take forever. Finally, we have the actual read from the sequencer again, and we have the base quality scores as seen on the FASTQ file. So although the SAM file is easily human readable, it is more demanding on a computer. Therefore, to save space and time, the SAM files are converted into what's known as a BAM file, which stands for Binary Alignments Map Format. So both these files contain the exact same data, but the BAM file is only machine readable. That is, if you try to vim the BAM file, you can see at the bottom of the screen there, if you try to vim the BAM file, you cannot interpret what these random characters mean, but the computer can. Furthermore, many downstream softwares also require the file to be in a BAM format to proceed. Therefore, to convert the SAM file to a BAM file, we use the toolkit known as Samtools. Samtools has various kits that will allow you to switch back and forth between file formats and also other helpful tools as well. So making the SAM file machine readable for easier downstream processing is great because it reduces computational workload. But time is still lost because the reads in the BAM file are, the reads in the SAM/BAM file are randomly distributed. That is, the list of reads we have can start from chromosome 1, followed by chromosome 17, then 16, then back to 1. So it's not in order. So by sorting the reads, we will further reduce the computational workload, which is required again for downstream manipulation of the data. So reads can be sorted by various attributes like name or tag value, but generally the default is to sort by genomic coordinate. So that the file starts with chromosome 1s and proceeds chronologically. So let's do a quick example. Hopefully okay, great. So the BWA is finished. So if we ls again, we can see that now we have a SAM file sitting there. So now we're going to do the same thing, but Samtools. So you would have to add it to path, which I've already done. So if you type Samtools, press enter, you have all your possible algorithms that you can use. For our case, we're doing two things. We're going to sort it. So you can see over here, right there, sort alignment files. And we're also going to switch to a BAM format, so view. So this can actually be done. So these two things can be done simultaneously. So let's do that. So we'll do Samtools, view. We'll start by converting to a BAM. So if you press enter, Samtools, view. You have all your options for Samtool views. We want to output a BAM, so we're going to use the -B flag over here. And before we continue, we also want to do Samtools sort, enter. You have the usage for Samtools sort, and we're going to do these together so to save time. So we're going to type in Samtools space view. We're going to use the -B flag. We use a -B flag. We're also going to use the -S flag just so that it auto detects the format. It just saves times. We're going to put our input SAM file. So if we remember correctly, that was my SAM file. And we're going to use this straight line over here. So the straight line is called a pipe. And basically all that says is that it's going to take the output from the first, so it's going to take the output from Samtools view, and it's going to use it as input for Samtools sort. So we're going to pipe that space Samtools tools sort, and you can see for Samtools sort we need to put we're going to use the dash small o so that it writes to file. So we're going to put dash o space, and we're going to name our output ERR, whatever the name of the sample, dot bam, and we're going to press enter. And so now Samtools is running. Unlike BWA, it doesn't have a bunch of scrolling things, but you know it's running because you can see this little spinning thing. So while that happens, let's get back to the presentation. Yeah, so far we've reduced the computational workload by making the SAM file machine readable, by converting to a BAM file and sorting the data. However, more can be done to further reduce workload, and this is through indexing. Indexing a genome serves to further facilitate the retrieval of sequences and their mapping. So let's take a book, for example. If we want to learn about, say, the environment, we go to the index of the book, we look under the letter E for the word environment, and we see all the pages on which we can learn about the environment. This is much, much faster than actually flipping through each page of the book and scanning through to look for the word environment. So this saves a lot of time. Similarly, if you want to find where in the genome a string of nucleotides go, we need to index the genome. Since DNA sequences are not words exactly, we take a certain number of bases, which is known as a k-mer, where k is the number of bases taken. So in this example, we are going to create a 5-base, we're going to index with 5 bases. So these are known as 5-mers. So when creating an index, you start with a string of nucleotides, which are used as a reference to make the 5-mer index, you start with the first 5 nucleotides and give it the position 0, or also known as offset 0. We then move 1 nucleotide across. So we started from C, now we're at G, and 5 from G, and that becomes offset 1. So that 5-mer is now offset 1, as shown here. And so you do this until all the whole string you've created all possible 5-mers. As you can see, at offset 4, we have a redundant 5-mer, so it's grouped with its partner in offset 0 and 4. Yeah. So these indexes are also ordered alphabetically to facilitate referencing it, or in other words, querying it. So let's see if we'll do that one live. So as you can see, the Samtools is finished. If we do ls, we now have a BAM file in our directory. So we're going to index this. So again, we're going to do Samtools, and we want to index. So and we want to index our alignment. So we're going to use Samtools index, Samtools space index, enter. So we have our options, and what we want is this BAI format. So this is the index format we want. So we're going to do again Samtools space index space -b, because we want the BAI format, space, we're going to put the name of our BAM file, BAM, followed by, yeah, no, just that. So you Samtools index -b, name of the BAM file, press enter. Now it's indexing. So back to the presentation. Yeah. So when we want to know where a sequence or read aligns to, we query the index using a 5-mer from that sequence. So we would take a 5-mer from this string. So this would be equivalent to a read that was found within our file. So it would take a 5-mer and it would query the index that was created. And we can see that the first 5 nucleotides here align to offset 3. After the offset is identified, the remaining nucleotide is validated against the actual reference genome to see if it actually fits. So by doing this, we make the computations much quicker, and this needs to be done for our BAM files for further processing and to visualize our BAM files. So the reference genome is also indexed. I've done that for you guys earlier in the package that I had included. This is a different kind of indexing, a much more thorough one that's also done through Samtools. So now that all these processes are done, we are ready to visualize or start visualizing our data. So let's just see if it's done. If I do ls, you can see we have the bam.by file right here. So to visualize, we use IGV. So this is IGV. It's from Broad Institute. It's freely available online. You'll have to upload a reference genome yourself. It's fairly simple, and there's many tutorials online. And so what we're going to do is we're going to drag that file that we had initially made. We're going to drag the BAM file, and what we're going to do is we're just going to drop it here. So right now we don't see anything because we're too zoomed out too far. So So the data set we use is actually from neuroblastoma and is specifically focused on TP53, the gene TP53. So let's zoom in on TP53. Just type in TP53, press enter, give it a second to load. So now you can see, if we can even zoom in a little more. So all these little bars are aligned reads to the genome. And you can see over here, these are your different exons. So you can go to various exons. And this is a whole exome seq and not a whole genome. So you can see where everything is aligned and what mutations are present by just okay. So it won't let me zoom in anymore for some reason, but it's more of a zoom thing, not an IGV thing. But generally, you can zoom in closer and closer, and each one of these colored bars that you see is a potential mutation. And I say potential because there's still a lot of filtering that needs to be done before we can claim that these are in fact real mutations that are present in the samples. So once we find these mutations, once you go through the pipeline and look at what mutations are present, and again, you can zoom in a lot closer on IGV and see exactly what the base pair changes are, how many times you see it. It's all present on IGV. But yeah, so once we've done these 2 steps, we have to go through the whole base recalibration, marking duplicates to get rid of any possible errors during sequencing. Once the processing is done, we can now use the mutation information and cross-reference online databases such as CBioPortal and other online large online data sets and databases for cancer, for example. That's CBioPortal, to see if there are any important trends that are associated with that mutation. So now I will let Robin elaborate on how to properly use online databases. Keep in mind there's going to be a cancer focus, but this can be extrapolated to other online databases that are available as well. So I'm going to my screen sharing.
Sorry, Michael. Can you help me with the screen sharing? Currently, I'm being disabled. Could you just allow me to do a screen share?
Yeah, sure. Should be working now.
Perfect. All right. Wonderful. So why might we want to compare our data with online basis? It's because typically our sample sizes will be very small. You might be working with a few animals or cell lines. So if you get something you think is interesting, it's very useful to compare your results with what other people have discovered. And typically those we stored on long-line repositories and databases. In cancer, the top resource that we all know and love is called the Cancer Genome Atlas, also called TCGA. And they collected thousands and thousands of cancer samples, and they collected petabytes of different omics data. So including genomics, transcriptomics, methomics, proteomics, and more. So we will use this as a case study to say, what are some of the benefits of having high quality databases? And we encourage you to think what kind of databases you might want for your work in synthetic biology. So I'll be doing a quick live demo. The website link is there, cbioportal.org. So let's say we have a scenario. We had the data set Ansley showed us. Our PI says, maybe there's something interesting about TP53 mutations and neuroblastoma, which is a type of childhood brain cancer. So we might want to answer a few questions. So I'll walk you through these. So maybe we want to say, how often is TP53 mutated in neuroblastoma? So now I'll switch to cbioportal. You can see it's a very pretty website. And here are listed all the various cancers that you might be interested in. So we have a search bar. So you'll want to type in the type of data set you're looking for. So it's found 4 different neuroblastoma data sets. I'm going to be going with this one because it has over 1,000 different samples. And keep in mind, these cancers are quite rare, so having 1,000 samples is really impressive. It's asking us what type of data are we interested in, and we said mutations, so we select the mutation data. Here you can custom select different patients, but we'll just say, show me all the patients in the data set. And here it's saying, what gene are you interested in? And of course we said, we're interested in TP53, and that's it. That's as simple as it gets. You selected a cohort of patients, what data you want to look at, and what gene you want to look at. So once you press submit, it's going to show all the patients who had something mutated in their TP53 gene. And actually, this was quite surprising when Ansley and I took a look, because TP53 is one of the most frequently mutated genes in cancer. And here, no patients, none in these over 1,000 patients had any mutations. So that answers question 1. So actually, TP53 is never mutated in neuroblastoma. I'll come back to question 2. So to say, well, why is TP53 doing something in other cancers? Why do people talk so much about it? We can ask cbioportal to say, okay, don't show me glial neuroblastoma. Show me a collection of all different cancers. So this is the MSK impact study. They have over 10,000 different samples. And we're going to do the exact same query, but with a different data set. So now it's just saying mutations in the gene TP53. And because there's so much data, it can take a few seconds to run. And it says, no, actually, TP53 is mutated in 42% of the patients here. And in fact, we can say, does this gene impact survival when you have a mutation? And there is a data set, a data visualization showing the survival of these patients. And when doing any sort of cancer or biological research, you have a phenotype you're interested in, and you want to see how your genotype influences your phenotype. So here under the survival tab, this is showing you what's called a Kaplan-Meier curve. On the x-axis, you have how long the patients have been alive since their diagnosis. And on the y-axis, it's the percent of survival. So everyone is alive at the beginning. And unfortunately, as time goes on, the survival percentage goes down. So you can easily see that the red line, which is the altered group, so patients with any sort of mutation in their TP53, their survival is much lower than patients who have unmutated or a wild type P53. So we know that P53 from this clearly is doing something to the tumors. A mutation TP53 makes the tumors more aggressive. That's what we would say. So now we might come back to say, well, we already saw that in neuroblastoma, TP53, it's never mutated. So it's not contributing to the progression of the tumors. So we might want to ask question number 4. So what might be the genes that are making neuroblastomas progress? What genes are important in this cancer? So I will go back to CBio portal. I can say, show me the neuroblastoma data set again. And it has a built-in tool to show a dashboard, a summary view of what's going on. And this is really useful when you want to explore your data. You don't really know too much about it. So there's a lot of information here. The one I want to draw your attention to is this particular little section. So this is called CNA genes, and this means copy number alterated. So what genes are the most duplicated or deleted? And duplicated means you have normally 2 copies of DNA, but in some cases you have 3 copies, 4 copies, so more copies than usual. And deleted means the section of DNA has been deleted. And we see that one of the top amplified genes in all neuroblastoma tumors is a gene called MYC-N. And it says here that MYC-N is a known oncogene, which means a cancer promoting gene. So now you might be able to go back to your PI and say, you know what, we found TP53 to never be mutated in neuroblastoma. We know it's a driver in other cancers, but in neuroblastoma, maybe it's a better idea to look at a amplified oncogene called MYC-N instead, because that's what we find. And all of this, you can make more hypotheses. You can go back in and dig deeper. Refer you to check out the tutorial section of the website.